This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 401, recorded on August 5th, 2016. This episode is brought to you by CuriosityStream, a subscription streaming service that offers over 1,500 documentaries and nonfiction series from the world's best filmmakers. Get unlimited access starting at just $2.99 a month. And for our audience, the first 2 months are completely free if you sign up at curiositystream.com/microbe and use the promo code microbe. This episode is also sponsored by Drobo, a family of safe, expandable, yet simple-to-use storage arrays. Drobos are designed to protect your important data forever. Visit drobo.com to learn more. Hi, everybody. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today here in New York City, Dickson Despommier.
Hello, Vincent.
Dickson, we are now on our way to 500.
We are.
401. It
Amazing.
seems so lonely, 401.
Well, that's because it's usually the sign you get on the internet when the website doesn't work.
That's 400, I think. It was 401. You're right. Dickson did a technological tour de force. You actually remembered. You must get a lot of 401.
I get a lot of 401.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here.
Where are you?
I am in my office in Western Massachusetts, where it's
standing up.
Yes, standing up, and it's comfortable in the office. It's a little warm outside. It's 85 Fahrenheit, 29 C.
Here too.
28 C here. We got these white clouds in the sky, right, Dickson?
We do.
Yep, nice puffy summer cumulus clouds. Yeah,
55% humidity. Also joining us from southeastern Michigan, Kathy Spindler.
Hi, everybody.
Hey, Kathy.
There are her dulcet tones.
Indeed. Maybe we should rename the group. And now for your listening pleasure, the dulcet tones.
Right.
With This Week in Virology.
So the weather here sounds about the same. 84 degrees Fahrenheit, 29 Celsius, and blue sky with white puffy clouds.
Nice.
Simpsons clouds, right?
Yes, Simpsons clouds. Yep.
Although I guess I'm the one who lives right near Springfield, so mine would be the Simpsons clouds.
Oh, I have to figure out where Springfield, Michigan is. I don't think I know.
There's a Springfield everywhere.
Exactly. Because there was a spring usually associated with that field.
Right, and that's where you built a town there.
That's true. The springs are gone, but the town is still there.
Springfield and Fairview, I think, are the two most common town names.
Yep.
We start with some sad news, the passing of Beverly Griffin. This was passed on to us by you, Kathy. Did you get it from someone? I don't remember.
Yes, I got it from Rosemary Rochford, who started out in polyomaviruses and is now working on EBV. So she overlapped with two of Beverly's areas of research, polyoma and Epstein-Barr virus.
She was 86 and a leading expert on polyomavirus.
Wow.
The first woman to be appointed professor at the Royal Postgraduate Medical School at Hammersmith Hospital in West London, and an example to aspiring women in science. This is from The Guardian. That's pretty good to have an article in The Guardian, isn't it?
You bet.
Mm-hmm.
Yes.
And there are also a couple of other articles that we have here, but Rich Condit weighed in. He's not with us today, or he's not here, I should say.
Please.
The other way is not right. He's still with us, but he's not here. He wrote, I did in fact overlap with Beverly at the ICRF for about 2.5 years. From 75 to 77, I was 27 to 28 years old. The entire fifth floor of the ICRF was polyomavirus, with labs run by Lionel Crawford, Bob Kamen, my official mentor, Alan Smith, Mike Fried, and Beverly.
Mike Fried.
He was freed up. I have to remember it that way. He was freed.
Indeed.
By the way, Lois Miller was with Fried at the time. Robin Weiss had his lab there, and Renato Dubecca was director. Dubecca won his Nobel during my time there. It was a rich, highly interactive, and productive environment. However, I remember Beverly most for the mentoring she gave me. In many ways, Beverly was my go-to person for any matter, both scientific and non-scientific, as I tried to sort out my future. She was always available with patience, wisdom, a gentle smile, and a twinkle in her eye. I benefited greatly from my time with her, as I'm sure did many others. I'm sad that she's gone, but thankful for the memories and the insights she shared with me. R.I.P. Beverly. There's a lovely thing here in, where was it? She was originally born in Louisiana.
Mm-hmm.
And she never lost her gentle southern U.S. accent. Did you ever hear her speak, Kathy?
No, I was never overlapped with her anywhere.
You never met her any meeting or anything?
Not that I know of. Was she at anything I was at?
Anyway, in the photo in The Guardian, she definitely has a twinkle in her eye.
Yes.
Yeah.
they did a good job with the flash.
Yeah, she does. Also in the one that's a Cold Spring Harbor photo.
Mm-hmm. Looks friendly.
Nice. Yeah, so she was instrumental in determining the sequence of mouse polyomavirus.
Right.
When it was completed in 1980 at 5,293 base pairs, it was one of the longest pieces of eukaryotic DNA ever sequenced.
And I think from that article it said she learned sequencing from Sanger, right?
Yep.
Not a bad tutor.
Not a bad tutor at all.
Right.
Now we have a follow-up from William who writes, Dear Professors, I greatly enjoyed TWiV 400 with Harold Varmus, someone I have long considered to be an inspiring figure. Throughout the course of listening to the episode, I had in the back of my mind a question I was told to expect during one of my medical school interviews in 1997. If you could invite any 3 people, living or dead, to dinner, who would they be? Since Harold Varmus was one of my people, for me the entire episode was almost as if a variant of that hypothetical situation was unfolding. I'm sure you can imagine how pleasantly surprised I was when Dr. Racaniello asked the very same question of Dr. Varmus. What person interested in science wouldn't love to be part of a dinner party, including Varmus, Enders, Rouse, and Thomas? What a conversation that would be. Looking forward to the next 400 episodes and more. Maybe in some future episode, the TWiV hosts can give their own responses to the dinner question.
Nice.
Bill is MD, PhD, Associate Professor in Pediatrics at Northwestern University Feinberg School of Medicine, which, of course, is in Chicago. Now, I had asked them one, right? Because someone told me this question a long time ago, and I used it in a couple of ASV TWiVs.
Right.
And it's usually 1, and then he changed it to 3. Maybe that's because what the medical school question is, right?
Yeah, 3 is better.
Why?
Because it could be really hard to narrow it down to 1.
Well, that's the point, right? You make it.
What one person?
But mine would be Einstein. So you guys can think about it I'm sorry, I said guys again. You people here on TWiV, my co-hosts on TWiV, all of you may think about it, or you can give an answer if you'd like. But mine's Einstein.
I'm torn between Darwin and Pasteur.
Yeah, that's a good choice.
But I don't know how much English Pasteur spoke, and my French is horrible, so probably I'd have to lean toward Darwin.
Good. We'll be having dinner together then, Dylan.
Do you want Chuck
good.
also? Yeah.
No, I love Darwin. I would love to hear what he had to say about his voyages.
How about you, Kathy?
Marie Curie.
But
good.
the French, right? Did she speak English?
I don't
But you
know.
don't speak French, you told us.
I don't know
okay.
French, and I don't know Polish. But in this hypothetical situation, that doesn't matter.
It shouldn't matter. You can have
Yes,
Marie
right.
Curie. Yeah, that would be quite an interesting conversation. You'd have to stay away from her so you wouldn't get irradiated.
Exactly.
I don't think she had any secondary emissions coming out of her.
But it's a joke to emphasize the fact that she was highly irradiated.
She was a glowing personality, yes.
She certainly was. Arg.
We have a little Zika sit rep for you. You did want it, didn't you?
Absolutely.
Well, I hope so, because you're going to get it.
Before the Varmus episode, one of our listeners tweeted, how much of the next episode will be on Zika? And I said, well, probably not much because it's Harold Varmus.
Right.
Yes.
Although we did talk just briefly about Zika funding, right?
Right, we did.
Now, as there is a page on the CDC website called Case Counts in the U.S., and as Kathy pointed out in the show notes, they don't keep it up to date. They finally updated it, I think, this morning to August 3rd, which is not even up to date, right?
Right.
And travel-associated cases in the United States, the 50 United States, 1,818, sexually transmitted, 16 Guillain-Barré, 5. So those 1,800 are all travel associated. They're brought in. People come in with virus and they develop illness here.
Allochthonous cases.
Oh, allochthonous?
Allochthonous cases.
Now, Dickson, are there some autochthonous autochthonous cases here
yes,
in the U.S.?
Yes, there are.
Yeah, so
they're clustered.
I think it was last week that the first locally transmitted cases were reported in Miami, Florida.
Yeah.
And what's it up to now, 16?
Not just Miami. It's in a specific region of Miami.
Specific neighborhood of Miami.
It's clustered.
Is in a specific neighborhood.
That's right.
And what is the name? Wynwood neighborhood.
Wynwood
which
neighborhood.
is a gentrifying area with restaurants and art galleries north of downtown. And every news article on this has a little map of Wynwood.
They mustn't be cleaning up their standing water. That's why.
Well, I don't think that it's specific. I don't think the mosquitoes are specific to Wynwood.
No, I don't think so either. They're all in Florida, right?
Right.
And it just so happens that this is the first place. So what likely happened is that someone, with 1,800 travel-associated cases, people are coming in regularly viremic, and someone came in, I suppose viremic, and was bitten by a mosquito, and then that mosquito transmitted it to someone else. Although, as I said to Dickson the other day, I guess it's formally possible that a mosquito came in, right, Dickson, on an airplane?
Or was blown in by the trade wind.
From
the southern region of, let's say, Cuba or some of the other
but I think
islands.
It's more likely that it's a person that's infected.
Yeah, it's
right?
it is formally possible is the right term. It could be that the mosquito arrived, but it is far, far more likely. Aedes aegypti is all around the southeast.
You're right.
And has been for a very long time. So this was, I think we've mentioned on TWiV, this was pretty much inevitable.
Yeah, of course, they're collecting mosquitoes, and now the reason why these are locally acquired or thought to be is because none of these people traveled to areas where Zika is ongoing, and none of them have had close contact with someone who's known to be infected. Although they could have had close contact to someone who is not known to be infected but actually is, right? We don't know that. Now, I understand they're collecting mosquitoes and looking for Zika virus by PCR, and so far all the mosquitoes have been negative. But at some point they should get virus or viral RNA out of mosquitoes. Right. That would prove it beyond a doubt, but it looks pretty good as far as I can tell.
And I would be seriously surprised if it remains confined to this one neighborhood of Miami.
Oh, right. Yeah.
I mean, they say in the article that in the summary on the Times, which has a nice what we know and what we don't know synopsis, that they're concentrating mosquito control efforts on that neighborhood. And I certainly assume and hope that they're continuing mosquito control efforts all over the area. Because it just happened in this particular neighborhood, and this is going to happen again in other places throughout the Southeast.
A lot of these outbreaks could be a catalyst for the reintroduction of medical entomology as a graduate school subject.
Don't count on it, Dickson.
Yeah, well,
but
because the paucity of information here is palpable.
Sure. Well, but there are people in public health who identify as medical entomologists.
Yeah, I know.
Mm-hmm.
They're just maybe not a formal course in most graduate programs, but people pick it up as they get into vector biology.
Is Bob Guadz a medical entomologist?
He certainly is. He's bona fide.
Bona fide.
Bona fide. In fact, the first laboratory in America to colonize a laboratory strain of Aedes aegypti was at Notre Dame, which is where Bob got his degree. With George Craig.
You did something there also.
Yeah, I worked with them in the summer before I came to Rockefeller and worked on Aedes aegypti. It's easy to work with.
Now, the question of whether it's going to spread, we can't really answer, but we can get a few clues from dengue virus, another flavivirus transmitted by Aedes aegypti.
Or chikungunya?
No, not a flavi, Dickson. Please.
No. I meant Aedes aegypti transmitted.
Also transmitted by, yes, we could get some lessons from that as well. Yep. But I have a paper in front of me from 2014, Origin of the Dengue Virus Outbreak in Martin County, Florida, USA, 2013. This is from Sharon Isern and Scott Michael down at Florida Gulf Coast University. It was the last outbreak of dengue in Florida. And this was 85 cases. But it was imported initially, and then it spread locally.
Sure.
This is Martin County is 275 miles north of Key West.
Huh.
I guess it's on the mainland, right?
It would have to be.
That would be.
So they had it's a pretty localized, I think, a single county and not many cases. And again, and since then we have not had an outbreak. So I suspect if it spreads, not going to spread a lot.
I think what's going to happen is Zika will go into an equilibrium very much like dengue, where you see it occurring throughout the Caribbean and popping up occasionally in Florida, where you'll have an outbreak surrounding one case that was imported, and then those cases get spread locally, but it won't go a whole lot further than that.
Yeah, so dengue has never spread locally outside of Florida or Texas.
Right.
The last outbreak in Texas was in 2005.
So far.
Right. Yeah, so far.
I mean, these mosquitoes, yellow fever used to be a huge problem throughout the Southeast, and they'd have huge outbreaks of it in the 19th century. So the mosquitoes are not gone.
No, of course not. But the way we live is different.
The way we live has changed dramatically, and people are just not exposed to these mosquitoes enough to really sustain that scale of an epidemic.
Yeah.
In this country, in the 50 states.
Sure.
It's different in Puerto Rico, where the mosquito population is higher and the climate is more tropical, and you don't have a hard frost to kill off the larvae ever.
Yeah, but Alan, we do surveys for mosquito species all over the place, and
right.
And Aedes aegypti is localized,
yes,
quite localized to the southeast.
Yes.
So even though we had yellow fever in New York City and Philadelphia, those conditions, the standing water and cleaning up the standing water, that's been the difference, I think.
Yes.
So the CDC.
Well, they're cleaning up the standing water, but also air conditioning.
Yeah, that does.
Indeed.
Also.
And houses that are airtight except for screened windows.
Sure. So I don't know if you know this story or not, but there's a big book written about the yellow fever outbreak in New York City, and the way it was cured, in quotes, was for everyone to go inside of a church and pray. And they actually stayed inside these churches for like days and weeks, and that broke the epidemic.
Right. Okay.
So the CDC has issued a travel advisory, which I understand is the first of its kind within the U.S.
Interesting.
For not to travel to this area. Is it? I think it's for pregnant women or everyone. I don't remember. Does anyone remember?
I don't recall.
Zika travel advisory.
Pregnant women.
Let's see. You think it's for pregnant?
At least from what I remember from hearing, I heard Tony Fauci on the radio last night. It was the same kind of thing.
We need to answer this, otherwise we'll get email.
Yes. Well, we don't mind getting email, but we do mind getting email about factual errors.
CDC issues historic travel advisory warning. They advise pregnant women and their partners not to travel to Wynwood.
Partners.
Yes, to Wynwood. Yes. Okay, so that's that. Historic, never been done before. And that's probably a good idea, right?
Yeah.
Mm-hmm.
If you have the option, if you don't already live in this neighborhood or have business there, and you were thinking about going to Miami, yeah, maybe a good idea. I mean, Puerto Rico is going to hate this because they've been really, really struggling to promote a tourism message. I get press releases from them constantly these days about how you shouldn't be afraid of taking your vacation in Puerto Rico because of Zika virus.
And it's a honeymoon destination.
It's a huge vacation destination for all sorts of things.
Yeah.
You know, this is really killing them at a time when they're already in very bad financial shape. So to have the CDC issue this warning for one tiny outbreak in a neighborhood of Miami, well, wouldn't you also issue a travel warning for Puerto Rico?
They have not, have they?
Not that I'm aware of.
Yeah, I'm just looking up here. It's not.
And also the neighborhood in Miami, I read somewhere some restaurant is way down in people going to the restaurant. So it's already affecting them for sure.
Affecting them, right.
Yeah, that's weird. All right. The other Zika newsworthy item is that the NIH has started or will start a phase 1 clinical trial for a Zika virus DNA vaccine. I think last time we talked about the paper, briefly anyway, describing the efficacy of a DNA vaccine against Zika in mice, right? Did we talk about it last time? At some point we talked about it.
Well, not episode 400, but the previous
399.
99, I think we did.
And now that was done by a group, among other places, at Harvard. And this is an NIAID developed vaccine, which is essentially the same. I think it's the two surface proteins, PRM and E, of Zika virus in a plasmid. And I don't know if they have any preclinical data on this. Because
they must.
They must, but there's nothing on their website and nothing in the clinicaltrials.gov announcement. Yet they're going into a phase 1, which is being conducted at 3 sites: Bethesda, University of Maryland in Baltimore, and Emory University. And this is going to have 80 patients, healthy volunteers, 18 to 35 years of age. Going to get different doses and regimens of DNA. And then we'll see if they have any adverse effects. And they will also take serum and see if there are neutralizing antibodies. So a 44-week total course. And I suppose at that point we'll know if they have neutralizing antibodies or not. And then, of course, if everything looks good and it's immunogenic, they could proceed to a phase 2. So that's like 2 months. So they could be in a phase 2 by the end of 2016, I would think.
If things go well.
Go well. Now, the cool thing about this page, we'll put a link to this page, where it's question and answers about the phase 1 clinical trial of the NIAID Zika virus investigational DNA vaccine. They also have at the bottom, is NIH testing or supporting research on other Zika vaccine candidates? And they have a list of what they're doing, which is really nice. They're working on it with GlaxoSmithKline, a self-amplifying RNA vaccine, and that's really interesting. It's in the preclinical stage. They're working on a live attenuated Zika vaccine based on the dengue attenuated vaccine that's being developed at NIAID, which we talked about some time ago. They're working on an inactivated Zika virus vaccine with the Walter Reed Army Institute of Research. And phase 1 trials are going to launch in the fall of 2016. And they are working on a VSV vaccine containing the glycoprotein of Zika virus, very much like the Ebola VSV vaccine that we talked about before. So they're doing a lot of different approaches, which is great. Yes. This is all NIAID, which is, of course, run by Tony Fauci.
Right.
And we heard a long time ago that he got together with all of his group heads and says, we're going to do this, and this is the outcome. I'm just wondering where he's getting the money for this. You know, he's getting some from Ebola because that's what happened, I presume, but it's expensive.
Right.
Yeah. Well, Rich had a conjecture that he put in the show notes. He thought maybe BARDA was somehow doing this.
Yes.
One thing that the live attenuated vaccine is already in a large phase 3 study in Brazil.
The dengue.
The dengue one.
Oh, that's right.
That's the dengue one.
Yeah.
Yeah, so that's one where they've attenuated the virus by making mutations in one particular, a 30 base deletion, I believe, in the 3' non-coding region. And this is all dengue. So it's not like the current Dengvaxia, which is yellow fever with dengue antigen. So it's all dengue. And we talked about this. It looks very good after one dose. So they're basing
it's a Zika vaccine.
So the Zika is based on that technology. So they're taking the backbone of that. They're basically taking the mutations that they put into that dengue vaccine and putting them into Zika. That's my understanding, yeah.
Well, because it's something, I mean, it's made it all the way to a phase 3 trial with dengue, and you're talking about a similar virus. So
I see. Okay.
Pretty promising idea, but they're just starting with the Zika project.
The assumption is that the changes will also attenuate Zika, right?
Yes.
Which they'll have to test in animal models and so forth. But
point of clarification, just for my own ignorance, what does BARDA stand for?
Biodefense Advanced Research something or
Biomedical Advanced Research and Development Authority.
Okay, it's what sprang out of the post 9/11 post anthrax attack mushrooming biodefense industry industrial complex thing.
Another source of funding could come from DARPA. That's what I was thinking.
Maybe.
We interviewed Rich Condit's buddy, who works in BARDA. What's his name? The guy from Buffalo. He's a cold room kind of guy.
Yes, right.
Is that Ed Niles?
Ed Niles, thank you.
Okay.
Talked all about BARDA. BARDA also, by the way, is a Marvel comic character.
Or
DC comic character. Sorry, not Marvel. Yes, there are all kinds of acronyms out there. Okay, Dickson,
Thank you. Yeah, that's
And of course,
makes me feel better.
As we commented last time, the Zika bill that would have provided more funding for this sort of thing didn't get through. But you know why? Congress is now on summer recess, and of course the reason they have a summer recess is because back in the old days, that was when everybody in Washington got vector-borne diseases, so they would all flee to the hills.
They don't need to do that anymore.
No, they don't actually need to do that anymore.
Just like we don't need daylight savings time, we don't need the summer off to help harvest.
Alan, you didn't mean to connect vector-borne diseases with flee to the hills, did you?
No, that was just that was
Thank
a convenient
you.
one on the way.
I just thought I'd give you credit where it wasn't due.
Right.
Good one, Dickson.
Fleeing to the hills. Yeah. This episode is sponsored by CuriosityStream, the world's first ad-free nonfiction service founded by John Hendricks of Discovery Communications. Over 1,500 titles. You can find it in 196 countries, and you could look at their content on the web and using these devices like Roku, Android, iOS, Chromecast, Amazon Fire, Kindle, Apple TV. And what they have is a wide variety of science and technology content. And they have ultra-high definition stuff, which is 4K, over 50 hours, one of the largest libraries on the net. What do they have? Things like Stephen Hawking's Universe, Next World with Michio Kaku, viruses. I have several shows on viruses. One of them is called Destruction and Creation, which talks about Zika. Life on Us, Microbirth, much more. With CuriosityStream, you get real science shows, not reality TV science, that you can find anywhere. There are monthly and annual plans available. They start at $2.99 a month, less than a cup of coffee, or one title on a competing platform. Check out curiositystream.com/microbe. Use the promo code microbe during sign up to get unlimited access to the world's top documentaries and nonfiction series completely free for the first 60 days. That's 2 entire months free of one of the largest 4K libraries around. Just go to curiositystream.com/microbe and use the offer code microbe at sign up. We thank CuriosityStream for their support of TWiV. And just to give CuriosityStream a little TWiV bump, got a letter from Johnny, our friend in Cambridge, who writes, went to curiositystream.com/microbe. I've passed the web address along to some colleagues who are parents. I can imagine elementary school science teachers might be able to find resources there. Thanks for a new and interesting site to explore. You sent a link to a video. Let's see what it is. What viruses, destruction, and creation. There you go.
Cool.
What is a virus? How does it affect humans? How does it spread? Yep.
Nice.
Okay. Thank you, Johnny, for looking at that. And we will move on to our paper, which was actually sent in by listener Trudy. Thank you very much for that. And it's published in the journal Immunity, which we do not often do here on TWiV, do we?
No.
Immunity.
No.
But you'll see this is quite relevant. It's called Host Inflammatory Response to Mosquito Bites. Enhances the severity of arbovirus infection. And the authors are Pingen, Bryden, Pondeville, Fazakerly. Did I skip one? Yeah, because they abbreviated. Schnettler, Cole, Meritz, Fazakerly, Graham, and McKimmey. They are University of Leeds, University of Glasgow, University of Tartu, which is in Estonia, and the Pirbright Institute in the UK. And this is it's not about Zika, but it's very timely, as you will see. It's all about mosquito bites and transmission of infectious viruses. How do you pronounce the first author, Kathy?
Marieke.
Marieke.
Right. Mm-hmm. So I can fill in bits of background here and there, but I want to point out that the paper is open access.
Yes.
Lovely.
Even though it's in Cell Press.
Uh-huh.
Now, the idea here is that, as we all know, mosquitoes take blood meals. Female mosquitoes take blood meals. And in so doing, if there's virus in the blood, they will acquire virus. And at the ASM meeting on Zika virus a couple of months ago, it was in June, I guess, I heard a lovely talk by Kristen Bernard, who is at the University of Wisconsin and who works and has worked with Laura Kramer, who was on the TWiV panel there. And they're interested in mosquitoes and how they transmit virus. And Kristen Bernard told the story of how mosquito saliva enhances West Nile virus replication in mice. So essentially, if you deliver the virus along with mosquito saliva, you get a higher viremia. And I actually did a little video on Virus Watch a couple months ago, and I actually included a movie that she had sent me of mosquitoes in her cage and so forth, and talked just briefly about how that was known, that fact. And it's been known for a while that basically when you put virus in by a mosquito, it enhances replication and disease. And it's the saliva that does this. You don't even need to bite. You just put saliva with your virus in and you get this enhanced effect. So this paper is all about figuring out what is going on. Okay, what is it about saliva that is enhancing? So we get more virus, and others have shown it can increase the pathogenesis of a virus infection.
One thing that's kind of fun from Kristen Bernard's paper is this finding that enhanced viremia was not observed when the salivary gland extract was inoculated at a distal site, suggesting that the salivary gland extract enhances West Nile virus replication by exerting a local effect. I think that'll come back.
And that will definitely come back, because you will see here it's a local effect for sure. So she was on the right track. So in this paper, they use two transmitted viruses, Semliki forest virus and Bunyamwera virus, two different families, and use a mouse model. So Semliki forest virus, for example, you inoculate subcutaneously in mice. Now, if you just put it in by a needle, no mosquitoes, the virus disseminates to the local lymph nodes. In this case, they're called popliteal lymph nodes. Within 3 to 6 hours, you get a low-level viremia. You get spread to distal tissues after 48 hours, and sometimes you get invasion of the brain. They have a attenuated strain that they mostly use for their experiments, and just occasionally it gets into the brain, whereas they have a virulent strain that just wipes everybody out, so that's less useful. All right, and what they think is that the virus is mostly replicating at the injection site. It spreads, and then possibly in the lymphoid tissue that drains it, and then spreads from there. So they do their experiments by allowing mice to be bitten with mosquitoes, and then they infect them right near the bite site with virus. Put 1 microliter of virus in. Okay, so you're separating the bite from the virus in that way. It's very cool, because you have to.
So the mosquito doesn't have the virus. The needle does have the virus. Yeah. You give the bite and the virus separately now.
Threading the needle.
Yes.
Yes, right. All right, so when you do that, you can show that the bite enhances the amount of virus that you find in the skin at the inoculation site and in the blood from 24 hours and onward, in the lymph nodes and in the blood. So allowing a mosquito to bite after you inoculate virus increases the viral load. And more of those mice end up having a lethal infection rather than an avirulent infection, which you get if you don't have a mosquito bite included. You see the same effect with Bunyamwera virus. It's different family, as I said, but transmitted by Aedes aegypti. And I don't know if I mentioned that these are Aedes aegypti mosquitoes that they're using here, and that both viruses are transmitted by that. So what do we know now? That at this early point, there's some skin, they call it a skin-centric process triggered by mosquito bites, has an impact on replication. So you get more virus, higher viral loads, and you get an effect, an increase on lethality just by having a mosquito bite near where you're putting virus. So that's quite interesting.
I don't want to sound like reviewer number 3 here, but it might be interesting to do this with a non-competent mosquito vector for these viruses as well.
It would be, sure.
Is it something special about Aedes? You know, is that part of the vector competence, or can you separate that out and use, I'm not sure what doesn't carry these two viruses, but if I don't know, Culex or something. Yeah,
that would be interesting. I
think that later figure kind of addresses that.
It kind of does.
But they're still putting in mosquito saliva with those or not? Anyway, I'm just looking at anyway, we can get to that later.
Right.
But,
all right, so what's going on here? They do a series of really nice experiments to sort this out. And I'm going to talk about them generally because it's a lot of immunology with protein markers that identify cell populations. I don't want to get into that because it's a little bit distracting from the results. But if anyone would like to know the markers, please, you can say them. But
and it's an open access paper, so any listeners who want,
if you really want it in detail, but I want
to dig into Figure 2.
I want to provide the overview. So one of the things they looked at was fluid accumulation at the bite site, because could this somehow influence transfer of virus to other sites? And so they have an assay for measuring vascular leakage. So what happens when you get any edema is the accumulation of fluid somewhere, and that's, in this case, of course, the mosquito is probing for a blood vessel. It's causing vascular damage, and there's leakage, and you get swelling and edema. And they see that 3 hours after a mosquito bite, there's a lot of edema in the skin. And, of course, you don't see this when you just put virus alone. And so they see an association of edema with retention of virus at the bite site. All right. And they kind of duplicated this by simply increasing the volume of the inoculum from 1 to 5 or half to 5 microliters. And they see that giving a larger volume also delays the spread of virus from the inoculation site elsewhere.
Yeah, so, I really like that. I thought that was cool.
I almost wouldn't have done it because I would have said, well, it's not going to work or something. But yeah, it's very cool. It's very straightforward. So the conclusion here is that the bite causes an edema that helps retain virus in the skin, and somehow this is facilitating infection, although we don't know how it's doing that. Okay. So next, how is this working, or what's going on? They look at what mouse genes are induced, innate immune genes, are induced by the bites compared to virus alone. And they see a number of genes that are upregulated, but they were struck by a couple that dominated the bite. Let's call this the bite-associated gene expression profile. A number of chemokines that are neutrophil attracting, and a number of other cytokines. And these, again, are not induced by virus alone. They are induced when you have the bite there. IL-1 beta is another one. IL-6, these are pro-inflammatory cytokines that induce inflammation, the influx of cells into an area. Also, they saw chemokines which are known to attract monocytes, like CCL2, 4, 5, 7, and 12. All right, so these are cytokines and chemokines that attract neutrophils and monocytes. So they were intrigued by this, and they looked by histology at the bite site, and in fact they did see an infiltrate of neutrophils from 90 minutes after the bite and onwards. So they could take sections and stain appropriately and look for this.
So neutrophils are a kind of phagocyte. They're normally found in the bloodstream, but during inflammation, that's when they're first responders and they migrate toward the site of the inflammation.
Right. And again, virus alone doesn't do a lot of induction of these neutrophil monocyte attractants. It's having the bite, so something that the mosquito is delivering is doing this. But they do note that the bite plus the virus does give you a more pronounced neutrophil influx than the bite alone. So what we've learned here is that neutrophil influx is a key part of what's going on here. Now, they wondered, does the virus simply replicate in neutrophils? So they looked at that, and in fact, they found that neutrophils are refractory to infection. So it's not a simple explanation that neutrophils are coming in, virus is replicating in them, and that's why you get higher titers. They wondered if there was a role for the adaptive response in this. They think that this is unlikely because it's all happening too early, but they did the experiment anyway. They take skid mice, which lack B and T cells, and they still get this bite enhancement of virus infection. So you don't need T cells, you don't need B cells, you don't need interferon gamma, etc. You don't need adaptive immunity in order to get this effect.
Right. So at this point, we know you need neutrophils to show up for the effect, but the neutrophils are not the ones getting infected.
You need neutrophils, and edema is also contributing.
And edema.
All right. They also wanted to know if the virus replication is suppressing innate immune responses. So that could be one way that you would get more virus So they look at a number of interferons and interferon-stimulated genes that are known to be antiviral, but there's no suppression. There's, in fact, enhanced induction of these, and I think that's because you're getting more virus replication. So it's not subversion of antiviral responses. It's something else. All right, so what is going on? The next step, they take away neutrophils. They deplete neutrophils using an antibody. And they notice that when they do that, the edema goes down, and that, of course, reduces or abolishes the bite effect on virus replication. So that makes sense if neutrophils are important. And what they're thinking about the edema is that the neutrophils are contributing to it. So initially you have some edema due to the mosquito probing, but then as the neutrophils come in response to whatever in the saliva, you get even more edema, and that potentiates the effect. One of the key genes that are produced by neutrophils is IL-1 beta gene, which is a pro-inflammatory cytokine. And this is important for inducing the inflammatory response at the bite site that gives this enhanced virus replication. Now, inflammation, of course, is the attraction of cells and the production of cytokines that come to an infected site. They look at whether, in fact, inhibiting inflammation by taking away neutrophils has any effect on virus replication. So when they deplete neutrophils, they reduce viral RNA production, both at the skin inoculation site, in the lymph nodes, and also in distal tissues. And viremia is also reduced to the point where it looks like unbitten controls. I like that, unbitten controls. Unbitten controls. There are a number of chemicals that are agonists that trigger neutrophil influx. So that's cool, right? And there are 3 different things that they can use that are unrelated: a 4-bol ester TPA, alum, and a ligand for toll-like receptor 2, PAM3CSK4. And all of these induce neutrophil influx. They induce IL-1 beta. And in fact, they enhance replication in the absence of a bite. So it's kind of duplicating what the bite does. So that's very cool. So far, mosquito factors at bite sites are somehow something in the mosquito saliva is promoting a neutrophil-dependent inflammation, and this leads to enhanced replication and dissemination. So there's some interest in this, whether you could have a therapeutic target, right? You don't want to get rid of neutrophils, but because in fact it turns out that they're important for protection from infection, irrespective of mosquito bites. But maybe you could modulate them in some way. So they look at an inhibitor of caspase-1. So IL-1 beta, which is a big pro-inflammatory cytokine produced by the neutrophils, is cleaved from a precursor by caspase-1. So they have an antagonist, a caspase-1 antagonist called ZYVAD FMK. And they give this to mice, and this results in fewer neutrophils at the bite site. No effect on virus titers, but inhibiting IL-1 beta production using this lowers virus titers at the bite site. So if you wanted to look into therapeutics, this might be a way to go. So that's a little side effect there. That's a little side trip, I think. So what's actually making more virus? So then they look into what cell types are going on, are involved in making more virus. And they have a green fluorescent protein labeled Semliki forest virus, which they use to infect in the same way they inoculate subcutaneously. And they ask, what cells is this infecting after the neutrophil influx, and you're getting all this enhanced virus? And it turns out that these are macrophages, which they can identify by surface markers and also histologically.
Right. So this is a point where Marieke told me that Stephen Bryden, who is the second author on the paper, it sounds like he was doing this as his rotation project before he joined for the PhD program, was to try and figure out what cells were infected, and they had hypothesized that the lymph vessels draining the skin would contain the cells that they were looking for. And so he used this particular marker that's on these lymphatic cells, but it also stains a subset of macrophages. And so the big surprise was that the infected cells were positive for this marker, but that they didn't match lymphatic vessels. Instead, they looked like macrophages.
Yeah.
So that was a really important piece of information that fundamentally changed how they were thinking about this. And then they started doing this more detailed experiments to identify that, yes, it was the macrophages.
Yeah, so they basically purify these cells using magnetic beads bound to an antibody to the particular surface antigen that marks these cells, and they put them in culture, and they say, yeah, they're infectable, and they release a lot of virus. Now, these cells, these macrophages, and other myeloid cells, which are included, they have a chemokine receptor, CCR2, and that's how they're attracted. Remember, CCL2 is the ligand. It was produced initially by neutrophils. So that attracts these cells via this chemokine receptor. And they take mice that lack the gene for the chemokine receptor, CCR2. And in this case, there's no influx of macrophages into the bite site. And there's no enhancement of replication. So you just take away that one chemokine receptor, the monocytes, the macrophages can no longer home into the bite site, and you don't get enhancement. And so that identifies those cells. Same thing, and it also works for bunyavirus, CCR2 dependent enhancement. So basically, what we have here, the bites enhance infection. They recruit neutrophils, which produce chemoattractants for myeloid cells, including macrophages. These get infected, so you have more infection going on than you would in the absence of a bite. You get a higher viremia, and you can disseminate to other tissues and promote virulence. Right. It's just gorgeous. Isn't it lovely?
It is.
And
it is.
I had to read the whole paper to really appreciate it, because when I first got it, I said, oh my gosh, look at all these facts plots.
That's right.
And I went through very carefully, and then after about a half hour, I said, I have to write a blog post on this, which I often do as a way of preparing for TWiV, and it just turns out to be lovely. And of course, there are a lot of interesting questions, but one of the things that I just wanted to point out, which I thought of, so I can imagine many, many years ago, there were viruses delivered by mosquitoes, and they didn't replicate in macrophages. They didn't do very well, because when you have a higher viremia, you enhance the possibility that another mosquito coming by later in that host will pick up virus and spread it. But maybe just by mutation of virus arose that could replicate in macrophages, and that is selected for, right? That's a powerful selection, because the selection is being transmitted to new hosts by mosquitoes. You have to have a high enough viremia in order to do that. So that's one thing that I thought is a cool outcome of this, thinking about how this was selected for. And of course, I wonder if, I'm sure they're doing this, if this happens with Zika virus as well, right?
Yes. Right.
That's where they're doing it. And since the paper has been out a couple of weeks now, many other people are doing it.
They're not the only ones doing it, yeah.
Right. So,
I wanted to add a little more about the identification of the cells being macrophages. So Marieke described probably her favorite moment in the exciting years working on this project, trying to identify the GFP positive cells and thus the virus replicating cells was a challenge, because she had to be able to get the cells out of the skin alive and then find the perfect time for when the virus is then expressing GFP, but before it starts killing the cells. And so she had to do these long experiments in the tropical heat of the insectary to get the mosquitoes to bite the mice, inject the virus, let the virus do its thing, and then for the perfect amount of time to obtain and stain the cells out of the tiny pieces of skin. And so it was well past midnight, and she says after about 15 hours of sciencing, the flow cytometer revealed a population of GFP-positive cells. I did a happy dance. So that's, if anybody's going to look at the paper, the happy dance is for figure 7B.
Well, in Glasgow, it's probably okay to work in a warm room, right?
I'll say.
Well, this is Leeds, but yes. Yeah.
Yes.
Leeds.
So in nature, if you grow up in an endemic area where mosquitoes are very common for most of the year, but there are seasons of dry and wet, but let's say the tropics in this case, it's estimated that little children get bitten about 1,000 times every day. So by the time they're 10 years old, They don't even know they're being bitten. I mean, you don't even see the marks.
They must get sensitized to the saliva component, right?
So that's what I'm driving at. What are those people like with regards to this infection?
Oh, because their edema would be much less.
Exactly.
Yeah, it's interesting.
Exactly.
So are you recommending, Dickson, that we all get bitten thousands of times?
Well, that's one way of desensitizing people, right? You desensitize with small injections of an allergen, and eventually you're not allergic anymore.
So here, that just leads to an idea for a study.
Yeah.
You take a population living in an area with lots of mosquitoes and a population where there are no mosquitoes.
There you go.
And you see the rate of infection with arthropod-borne viruses, right?
Yeah.
Interesting.
Yeah.
Yeah, because 1,000. I've heard that number 1,000 times a day.
Yeah.
So at some point they probably don't even have the bumps, right?
Exactly right.
Right.
No neutrophils coming in.
Yeah. I'm going to post a picture of a friend of mine.
Where are you going to post it?
I'll give it to you.
I see.
You can use that as a show notes as to what it looks like for a naive host to get bitten for the first time by an arthropod. And in this case, these are little no-see-ums that are common throughout the Caribbean. This was in the island of Andros. And we were fishing for bonefish, and he was walking along the shore with his guide, and his both arms were exposed, but only his left arm got bite because the right arm was facing out towards the ocean. The left arm was facing towards the shore, and the little midges that could barely fly encountered the left arm, and his arm was, it looked like a shotgun had the splatter.
Wow.
It was just awful. He was up for 3 or 4 nights. He had to take lots of codeine. He almost went to a hospital.
So, Kathy, how do they get the mosquito and the virus at the same place? Do you know?
Well, so I wrote to Marieke because I wanted to know more about this. They have pretty good description in the methods and then in the expanded methods of they want to infect just a very small part of the mouse so that they know exactly where to stick the virus in.
Right.
And so she sent a picture, which is really worth 1,000 words. So they have these, the mosquito cages have a netting on top of them, and they cover a large part of that net, and that's where they put most of the body of the anesthetized mouse. And then they extend a portion of its foot and tape it down, and they also put tape over the toenails. And so there's just a very small amount where the mosquitoes can then come up and go through this netting and bite. And they count for 1, 2, 3, 4, 5 mosquitoes to bite, and then they take the mouse off and they inject into that site. Fantastic. And so it's more than a one-person job to be counting and then get ready to inject.
That's amazing.
Kathy, in this picture, they're like 10 mosquitoes.
Well, really, she says you can see 4 engorged ladies in a small area around the foot. So I think we have a little problem with parallax, figuring out exactly what's, going after the foot.
Looks like some of them are trying to get to the body, which is covered.
Yeah.
Right.
It's really impressive.
Well, because they sense the heat.
Yeah.
Yeah. Right.
So they're going and probing where it's warm, but those are just getting paper towel or whatever that is, or bent paper.
Aluminum foil or some kind of foil.
Oh, okay, aluminum foil.
Foil and fabric. Yeah.
I can see a few of the engorged females there.
Yeah. I guess it would be aluminium foil.
Aluminium. That's right.
Another anecdote along these lines is that I was privileged to be at Notre Dame when they began their research on the Aedes aegypti, and the feeding mechanism was, in the beginning, they were using mice. Then they determined they got a better yield of eggs if they used human blood. So the director of the laboratory, George Craig, would roll up his sleeve every week, and he would bring in cages of female anopheline, of Aedes aegypti, and he would expose himself, just like this mouse.
My grandfather used to do that all the time.
And you know what? After about 2 months of this, he didn't have any red marks on his arm whatsoever. Totally desensitized.
Kathy, she writes, the mosquitoes were really keen.
Yeah. And this is, by the way, if we put this picture, I don't know if we'll put this picture up, but it's an anesthetized mouse.
Right.
Yes.
So otherwise it would be leaving.
I would like this one with all the mosquitoes. Can we use that, Kathy?
I think you check with her, or you could at least put a link to it. Yeah,
because it's really cool.
Yeah, or you just see part of his foot. Yeah. And she calls it a particularly bitey batch of mosquitoes.
Bitey batch. I guess there are some that are not as bitey.
Yeah, if it's a bitey batch, they'll give it 2 mice per cage so that they don't overfeed. There's some variation in the level of enthusiasm of the mosquitoes, but we always do internally controlled experiments to prevent that from skewing the data.
This is multi-systems research at its best.
It is. It's
great. I love it.
Yeah.
I just love it. You got the whole story.
Yeah, it's not just virus and cell culture.
Now you've got the whole
thing. Kathy, what's with the ears here? What's the relevance?
Oh, she just sent, sometimes they do the foot, sometimes they do a shaved back, and sometimes they do an ear. So here you can tell which ear got exposed to for mosquitoes.
Wow, look at that
vasodilation.
Yeah, it's beautiful.
And it makes it very clear where to inject the virus if you're doing that experiment.
Yeah. So the other thing I did,
nice.
I even read everything. Come on, I'm on board today. I'm very excited about this.
You mean you don't usually?
Well, sometimes I don't read everything, but I sent you 2 papers that did the molecular biology of the secretions of the salivary glands for Aedes aegypti.
How many proteins are in saliva?
They claim there's about 130 of them.
Oh.
But there are a lot of shared proteins with the males. And so then you have to subtract the male from the female to find out which ones are probably important for these
that's maybe
effects. And so there are about 30, they think, that are important for these bioactivities that all coordinate together to give the female mosquito a clot-free blood.
So are you saying that if we could identify a single protein that did all this, we could make a vaccine that would protect against all?
Well,
and assuming these are conserved among mosquitoes?
Well, that's the other problem, because the answer is no, they're not conserved.
Right.
Aedes and Culex are about the same, but Anopheles have different. They have different proteins that do the same thing. Yeah. So they arose at different times in evolution.
Not surprising.
Right. And there was a convergence. And
very cool.
Well, the blood feeding behavior probably arose at a particular time, whereas others were fruit feeders or something.
That's right.
I just noticed something, skipping back a few seconds to this mouse ear shot. It really nicely illustrates how precise the mosquitoes are, because you'll notice that all 4 of the bites are on little capillaries.
Absolutely.
There are none in between. They just all bang, 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 bang.
Well, they probe till they find
exactly
one,
right. But that ear really just illustrates.
So it's a heat-seeking missile, basically.
Yes.
There's a video that came out in PLOS One some time ago, which they videoed mosquitoes probing mouse skin, and you can it's very creepy, because you see the skin transparent, and all of a sudden this brown proboscis is coming in and out. It's just so weird, in and out. And then they find something, and then the whole vessel collapses because it has such suction.
I'd like really to redefine what you just said, because it's actually called a stylet.
A stylet?
Yeah, the stylet part of the proboscis, because there are multiple parts.
Yep. There are a lot of parts, right? Six or something.
Many different parts.
Pieces parts.
Yep, parts. All right. Anything else you want to say about this, Kathy?
Well, I wanted to just give a little more background about Marieke. Yeah. She had done her PhD in the Netherlands on HIV and drug resistance. And she had gotten kind of turned on to science by seeing some Discovery Channel program about HIV, drug resistance, and the CCR5 Delta 32 mutation. So that's kind of how she got her interest in science, and she did her PhD in the Netherlands, and just as she was celebrating getting her PhD, she found out that she had gotten this position to work in McKimmy's lab. And I think you're right. I think she first started in Glasgow, and then they only moved to Leeds as of February. And she has good things to say about Clive McKimmy. He's continuously optimistic. That's very contagious and motivating. And she said, you have the very best supervisor when they leave surprises on your desk, such as a muffin to keep you going on a long day.
That's good.
So, yeah.
Right.
I don't do that because I'm not allowed to put food in the lab.
It's too bad that she didn't record her happy voice and put it up on YouTube. That would have been viral. It would have gone viral, no pun intended.
Right. So, and then I asked a little about, what she wants to do in the future, and she enjoys doing virology as a scientist, so she'd like to continue doing exciting research on mosquito-transmitted viruses and the mammalian host response.
She's got a bright future.
Cool.
She's got a bright future.
Lovely. All right. Thank you. I want to tell you about our sponsor of this episode, Drobo. Welcome back, Drobo.
Welcome back, Drobo.
Hey, Drobo.
In a few years.
Who loves you?
But Drobo is back, and Drobo is a hard disk solution for your computer. It was designed to solve two problems that affect hard drives. One, they get full, and two, they fail. They're mechanical, unless you get a solid-state device. But we're talking about the mechanical ones, which most people still have. Hopefully, they happens in that order, full and then fail. And Drobo solves these two problems. It is expandable storage. You can buy different units with different numbers of bays for hard drives. A couple, 4. They have a 5. They have, I think, an 8. They have a 12 bay. Wow. Could put all the Microbe TV episodes on that one. And they use a proprietary type of RAID. RAID is a way that you can put multiple drives together and have them appear as a single drive. And Drobo has their own way of doing it that removes all the limitations. You can use different size drives. You can add drives at any time, and they just configure really beautiful.
And an important feature about RAID is that it's redundant.
Yep.
So a RAID array, if you have 3 drives in there, it'll copy multiple copies of your data onto the 3 drives and also give you additional capacity.
Right. So on these Drobo arrays, you have 5 drives in there, and one of them fails. You just pull it out and put a new one in, and all the data is copied from the other one. Well, the data are copied, and you can, I think, have up to 2 drive failures at once.
Yes. Very
cool. And the Drobo knows how much data are on the drives, and they give you lights. There are lights in the front that turn from green to yellow to red when you have to add a new drive. You like that, Dickson?
I do. The Drobo you gave me, I still use. I used it this morning, in fact, and I always have a good time just looking at it.
Yeah, this from the last set of...
The blinking lights are gorgeous.
They are.
The last set of ads we did, you got a Drobo out of that, Dickson. That's right.
I used it to store my visuals.
And it's very simple. So if Dickson can use it,
anybody.
It must be simple.
You got it right.
That ought to be their new slogan.
Dixon,
Absolutely.
if you can understand a traffic light, you can use the Drobo.
There you go.
It's red, green, or orange. They have lots of models. They have 8 products, and it depends what you want to do. If you're interested in photography, there are certain models. They're called the 5DT or the 5D. They have high-speed connections. If you want to take one out in the field, if you're a filmer or a photographer and you want to bring a big storage unit in the field, they have portable units. They're called the Mini, Drobo Mini, and it weighs 2 pounds. If you need a lot of storage, like to store all of the Microbe TV shows, they have a 4-drive or a 5-drive. They even have bigger ones as well. And if you use an Apple machine, you could set it to be your Time Machine backup. And use it for backup or whatever you want. And the cool one is the network Drobo. It's called the 5N. You plug it into an Ethernet port, and it's available on your network. So if you're at home, you can plug it into your network, and all your computers can reach it. Now, Drobo was kind enough to send a couple of us units across podcasts, actually. One here, a couple here on TWiV, one on TWiM and TWiVo. And as we do ads on the different shows, we're going to hear people's experiences. Now, I haven't set mine up in the lab yet. I got one sent to you, Alan. Did you do anything with it?
I got it, and I had bought... Last time they were sponsoring the show, they offered us discounts on Drobos, and I bought one.
Mm-hmm.
And I had set that up. That was 3 years ago or thereabouts. And... I will say that this is a beautifully boring experience once you've set it up, which is exactly how a backup solution ought to be. You set the thing up. I've got it sitting in the back corner of my desk, and it just sits there.
Yeah.
And you can, if you're a Mac user, you can coordinate it with Time Machine. I wrote my own little bash scripts to take care of it. It'll any system that you have for copying stuff, it just treats it like a mass storage array. You slide in a couple of hard drives. I think I had 2 terabyte hard drives, a pair of them or 3 of them, and it just sits there humming away. And you back up your data on whatever schedule you do that on automatically if you like. And then fortunately, knock wood, I have not had a hard drive failure since then. That's kind of how backup solutions often affect things, I think. But I know that if something goes on my main computer, I've got all my stuff backed up on my Drobo. And if one of the Drobo drives dies, it'll give me a warning and a red light. and I can just slide that out and slide a new one in. So just really great peace of mind and a reasonable piece of gear. And when the new one arrived, it's the same size package as the old one. So if you're upgrading from an old one, it's not a big deal. It takes up the same space. It actually uses the same power supply. I had it set up in about 15 minutes and transferred everything over with the same scripts. And it's just, in its new role. I have it turned off during TWiV so that it's not humming in the background. But
so you have it on your network at home, right?
I have it on the network. This one is this, what is the model? Is this the Drobo S or was the Drobo S? The 5N, that's right. Yeah. The previous one was the S. And I have it just plugged in to Ethernet into my router.
You can see it throughout your home, right?
I can see it anywhere on the network. I can access it, and then I back it up. I back up the computers from there. And I will add that if I had not gotten a free one, I'd still be using the old one. This is the type of device that you buy and you probably keep for 5 or 10 years at least. And there's not going to be, if you want to upgrade it, you can just slide out the drives and slide in bigger capacity ones.
Yep. Yeah, my use case is in a lab. I want to be able to share data among people in the laboratory, which can be done now, but it's not as easy. You have to sign into someone's computer over the network. It's never straightforward. But this shows up as a device on the network, and people can put all their images, data, graphs, whatever, and share it. So as soon as I set that up, I'll let you know how that goes, because I think the science is a good use case for a Drobo.
Definitely.
Anyway, if you're interested in purchasing a Drobo, you can go to drobostore.com. TWiX listeners can save $100 off their purchase of a Drobo Mini, 5D, 5N, or any 8 or 12 drive system. Drobostore.com. Use the discount code Microbe100. We thank Drobo for their support of TWiV and Microbe TV. Thank you very much.
And Drobo is spelled just like it sounds, D-R-O-B-O.
That's right. All right, it's time for some email. The first one is from D. That's all. No name, just D.
D.
And he writes, VR et al.
Because, of course, D would call you VR.
That's right. Although viruses are not specifically mentioned in the following article, it is intriguing to consider them and their assembly/mutation while reading. And this is an article in... no, that's not the right one. This is an article in Nautilus, actually, which was my pick years ago. Mm-hmm. Cool online magazine. How do you say life in physics? By Alison Eck, and it says a new theory sheds light on the emergence of life's complexity. And this is all about Jeremy England, who I've heard of. He's a young assistant professor at MIT. He has a new theory called dissipative adaptation, which is used to explain how complex lifelike function can self-organize and emerge from simpler things, including inanimate matter. And this is all about language and what words mean in different fields. So I can see where, if you're thinking about viruses, it would be interesting to take this in the same context. So it's pretty cool. Mm-hmm. So check it out. It's a nice article.
Cool.
Let's see. Kathy, would you like to read?
Sure. Niraj writes, Dear TWiVolutionaries, greetings from the sunny Bay Area in California. The last few days have been quite hot out here, but I'm not complaining. It's given me the opportunity to pursue outdoor sports and take my 1-year-old son to the parks, which he absolutely loves. On a more personal note, I've been an active TWIP, TWIM, and TWiV listener for a while now and was introduced to this fascinating world of most educational podcasts by my boss, Jeff Fairman. He was the only one who got the diagnosis right on your last case in TWiP, where the lady was observing moving object in her eye. And this happened when they were at a small startup called Sutravax in South San Francisco. I've been immensely enjoying listening to your podcasts, and so much so that I've begun canceling my music subscriptions to make time for listening to the fascinating world of parasites.
Dulcet tones.
Which never ceases to wonder. I began this journey of scientific research by graduating from Rockefeller University. I'm sure
Nice.
Drs. Racaniello and Despommier will agree with me that it is still a hotbed of fabulous cutting-edge research. Yep. And then I moved to the West Coast to pursue my postdoc work at Genentech as I wanted to experience the world of biotech. In reference to TWiV 385, and in line with your discussion with Dr. Firestein, I agree that the present climate for promoting young scientists to pursue academic careers is becoming exceedingly hard. And even though funding is an issue, I don't think it's the only major one. I can speak from personal experience as well as from the experience of several of my colleagues at the time at Rockefeller. Coming from a background in chemistry with no experience in biomedical research, I must say that my overall experience of learning lab science at Rockefeller was absolutely amazing. Mm-hmm. However, the most disappointing and shocking aspect of my stay was the attitude of my PI towards mentoring and cultivating an atmosphere of curiosity-driven science. Even though he gave me freedom to work on projects I was interested in, his inability to value time along with the ability to procrastinate were hugely demoralizing for me. Even after I finished work on 2 of the 3 projects I was working on, I had to wait for eternity, for God knows what reason, to submit the manuscripts that had already been written and edited so many times that I had lost all charm in getting the story out. My passion for science and getting the data published were always at conflicts with my PI's disinterest in getting the story out on time. On several occasions, I was reminded that he has a tenure track and one more publication wouldn't make a big difference anyways. Yeah, to his career, but for a young scientist like me, that was devastating to hear and kept getting harder and harder to still muster enough motivation to keep slogging away with the hope that one day this will all be over. This experience always made me think, why is tenure track really allowed? Why are these professors not accountable for their work like everyone else? Professorship is not about the comfort of the position, but it's a huge responsibility to cultivate the enthusiasm and curiosity of the next generations of scientists. Given that no one ever teaches a lab head about how to be a good lab manager, I think this is an aspect of that should be definitely addressed, since you can't let people have autocratic power without accountability. Why not have a review system where the lab members can anonymously vote and spell out their grievances? Isn't there a need for a review process right at the source where science is being done? This could also avoid so many mishaps and altercations that I've witnessed in my career. Still, the flame kept burning and the passion for scientific endeavor was as strong as ever. Hence, to pursue this, I applied and moved to Genentech for my postdoc. And the big change at a business enterprise was that everyone was accountable. No exceptions. It didn't matter who the boss was, but when it was about getting the work done, nothing else could come in the way, especially not your ego. And this was a huge paradigm shift for me to see what makes us a successful enterprise. The drive with which people attack a problem is infectious, and it's this culture that makes Genentech a front leader in getting breakthrough therapies approved day after day. I think your discussion with Dr. Firestein hit on several of these nodes, but without any active decision-making. I fear these will always be topics for coffee table discussion only. I've become totally addicted to these podcasts and would personally like to thank you for the noble effort of your entire team to have the passion for learning and educating everyone, because we know more when we all know more. The world of science is fascinating, and I can never imagine doing anything else but be at the bench and make an experiment work. The satisfaction is hard to describe, but the joy is immense to experience. We still have a long way to go and learn so much that's unexplored out there. Your series of podcasts are an excellent way in getting head start in feeding our curiosities. I wish you the very best, and Alan, you have a gift at coming up with catchy headlines. Contrary to what people have been telling you, Dr. Racaniello, your exchange with Dr. Despommier is most fun. I don't think you are mean to him, but just a very close friend of his.
Hear, hear.
Best.
No, I agree. You have no idea of what Vincent just did. He absolutely issued a friendly gesture with one of his hands to me as a way of solidarity.
He raised a finger to you.
He gave me the peace sign. Well, actually, he gave me half of a peace.
Half of a peace sign. Yes.
Half a peace sign. That's right. That's a good way to put it, Dickson. Boy, you're on a roll today.
That's not my line. That's a line from Dick Cavett.
Really?
He says, New York's such a friendly place. He says, I've only been in town a week, and I was hailing a cab, and the cab drove right on by me. He says, but he was very nice about it. He gave me a peace sign. Well, he gave me a half a peace sign. So that's what that comes from.
So on the tenure track review thing, where I was before at the University of Georgia, there was a 5-year review for faculty members.
Yeah.
And I'm not sure that it took into account comments from trainees in the lab, but there was at least some review.
I mean, the point here is that when you're tenured, you still have an obligation to train people if you're going to have them in your lab, right? So you have to publish their papers. That's part of it. If you don't want to, I mean, this attitude is absolutely absurd and not uncalled for. Yep. But unfortunately, there are people out there, and there's not the only one at Rockefeller. There are others. But my view is, if you don't want to train people and give them the right atmosphere, let them publish. Don't have people in your lab then. Figure out how to do the science another way.
Right.
And all of Rockefeller isn't like that, obviously.
Of course.
Well, and there's people like that at other places. It's not just Rockefeller.
Of course.
You bet.
But I'm really glad that he or she, I don't know, do you know?
I think it's a male name, but I don't know either.
Anyway, I'm glad that the experience reversed itself at Genentech. That's great.
Yes.
You know, and I think in industry, it's a little, he or she is right. It's you're accountable, although you can find bad people there as well. So I'm glad it worked out. Yeah.
And also, it varies. It's going to vary from company to company and place to place and individual to individual. Some people just really are much more suited to a corporate environment than they are to an academic one.
Yep. Absolutely.
And it's great that Niraj has found a home.
Yeah, it's really good. All right, Alan, can you take the next one?
Sure, Bill. Right. So he gave Kathy this 2-page letter, and now I'm going to read one that's full of links.
Just a bunch of links, yeah.
Yeah, you could take a couple.
Okay, I'll take a couple of them. Bill writes, in light of the brouhaha around the money mines of Elsevier et al., and sends a link to an article published in Nature's news section about the beginnings of peer review and how things were not all hunky-dory when the peer review system was first produced in the 19th century. Good article and free online, so you can go catch up on that. So some of these problems are not new. And then Bill also says the company that's upsetting their Apple Card, and sends a link to Sci-Hub, which is actually not a company, it's an individual. And there's an article, I don't know if Bill provided this or if somebody else did.
Yeah, they're all Bill's, yeah.
Okay. It's an article on Big Think, which I guess is a news site.
And I've been on Big Think.
Okay. It's about this anonymous researcher who set up Sci-Hub and made all of the scientific literature freely downloadable. It's like Pirate Bay or what was the one before that? Napster. But for scientific papers. So you can go and download these, even though they're copyrighted and behind paywalls. This researcher has made them freely available and circumvented all of that, which of course is totally against copyright law. But that's why it's an anonymous researcher doing this
But not any longer. If you read this Big Think article,
yes, not any longer, because that did eventually get shut down.
Yeah.
But it was it was an effort to, I think, advance this conversation a little bit faster. I'm not sure that I really endorse the approach, but I understand the sentiment.
I don't think Harold Varmus would have agreed with that either.
No, it's too inflammatory. Anyway, you could say it's an act of civil disobedience, which I'm not sure helped. And then Bill also links to a video from Piled Higher and Deeper Comics, which is talking to a couple of people, Nick Shockey and Jonathan Eisen, are talking about the world of open access publishing and how it works.
Yep.
So if you want to catch up on that, that's a good video to check out. Right, then Kevin writes, "Hi, TWiFolk. I have also run into objection to the term guys in the context of trying to be inclusive in the classroom. There are already so many subtle obstacles to female students doing well in science," and links to one of the many papers about that. "Because of this, even if the reason for the objection is baffling to us privileged white men, it's incumbent on us to remove what obstacles we can." Hear, hear. "I applaud you for your efforts to stop saying guys when referring to a mixed gender group, but you definitely need an alternative that serves the same purpose to make it workable. I wanted to offer one that's worked well for me. Folks is gender neutral, still one syllable, and doesn't have the awkward regionality of y'all." Hey now, y'all. That ain't awkward. "I've been mostly successful at replacing guys with folks in front of my students." And I agree, folks is... I start off emails to multiple people that way. I'll say, hey, folks.
It's too close to another word.
Well...
And I'm going to mess up, and I don't like folks. I don't know.
Yeah.
I don't know what to do.
Gang. Use the word gang. Hey, gang.
I have no problem with you all.
Yeah,
Or people. Listen up, people.
Hello, everyone. I'm Vincent Racaniello.
Hello, everyone. Hello, everyone. Yeah, that's pretty formal, but not bad.
Kevin is from Audio. How do you say it, Kathy? Audio immunity.
Audio immunity.
Audio immunity.
Audio immunity.
Right.
Audio immunity. Okay. Do you want to take another one, Alan?
Oh, sure. I'll take another. James writes, this is about virus dispersal by hand dryers that we talked about a little while ago. Greetings, Twivers. Great podcast across the brand, as always. Just getting back to them after my relocation from London back to Scotland. I'm looking out my window on a usually clear sky. No, it must be unusually clear sky just outside Glasgow with sunny 8 degrees C. It's not a usually clear sky just outside Glasgow.
Nope.
I had already come across the virus dispersal article, but I felt I had a couple of points to add. First, science communication with the public is a key challenge for science. I felt, however, this article was poorly reported to the public across all media, even with the summary of the article. Unless the public really thinks about this and reads the original rather than relying on other outlets, it isn't clear we're talking about dispersal from virus-loaded sources rather than post-cleaning. While I accept the comments about public facilities and a lack of real effort to clean, does the potential dispersal factor not just make it similar to any other fomite, such as door handles? Given the relatively high exposure this was given in traditionally non-science, high general population exposure, non-science, I think he means publications, more have been made more effort have been made to highlight the need for good hand hygiene. Right. So the idea is these hand dryers, the Dyson hand dryer and all the bazillion different knockoffs of it that use high velocity air. Apparently, according to this study, they increase the number of viruses you're blowing up into the air. But the question is that if you wash your hands properly and thoroughly, is that still the case? Or is it just the case when people do the kind of routine where they wave their hands in the general direction of the sink for show and then go over to the hand dryer, which is a good point. Second, given this was about dispersal, I've often had a thought about the paper versus hand dryer issue. Maybe this is something partly due to my UK upbringing and our apparent disproportionate interest, as Kathy mentioned. Paper towels have other drawbacks I have yet to see addressed. I've seen many times paper towels left within the dispersal zone of toilet facilities, placed near taps, soaking and likely absorbing bacteria. Another issue demonstrated in schools in Scotland through science experimentation is that a paper towel, when it comes in moist contact with a hand, will allow migration of bacteria through the stack of paper towels. So when you remove the paper towel with your wet hand, the one behind it is also damp. Just how often do bacteria pass on? This would be interesting to see weighed against the hot air dryers. What if the person before you has the beginning of a highly communicable disease? But most of all, with good hand hygiene, the air dryers would not be a significant problem, or at least no more than door handles and taps. Thanks for the great discussions and great guests. Okay.
Well, I must say that after having discussed that, every time I use one of those things, I think about it.
Right. How many viruses am I blowing around the room?
I'm fine. I think
how bad they are for your ears because they're so loud.
Yes.
Especially when there are a bunch going at once, right?
If there are paper towels available, I almost always use paper towels.
Yeah,
I feel badly about that. If you go to a restaurant, for example, and it's late in the evening, and the trash can is just overflowing.
It's true,
and that's just one restaurant out of hundreds. And oh my gosh, it's so much waste.
A lot of trees.
Wow. All right, Dickson, can you take the next?
Ken Yeager writes, "Dear Vincent and Dickson."
What's with the Vincent and Dickson?
Firestein.
Firestein, you and I did.
Because we were there.
Yeah, sorry.
And the others were not.
Okay.
Thank you for the wonderful interview with Dr. Stuart Firestein. It covered a lot of ground and was really thoughtful and thought-provoking. After the episode ended, I added both of his books to my reading list. I want to comment on the topic of incorporating history of science into the science class. I think it is a critical part of education, and the human stories of perseverance and failure and amazing insights must be told. However, I do have a small note of caution. From my recollection of myself as a student and from my limited teaching experience, I feel that students are not very interested in history. You may argue that I just was unlucky in not having a good teacher who could tell those stories well, but I think there's more to it. First, one needs to have at least a general picture of the present before the past can make any sense. You need to teach about the key roles of DNA in biology and or about the key questions of heredity before the student can appreciate the true importance of Watson and Crick's discovery. Alternatively, you would have to properly set the stage and describe the state of science at the time of discovery. Good approach, but it takes a lot of time. Second, when students begin to learn a scientific subject, they are presented with a long list of wonderful and amazing discoveries, which distorts their scale of progress. Everything is great and important in a breakthrough. One needs to see what a more typical discovery looks like and how the progress is made day to day in science in order to better appreciate a truly elegant and brilliant experiment. I found that my own interest in history of science and history in general grows in proportion to my age and knowledge about the world.
I have noticed this generally.
Yeah, exactly.
The older someone gets, the more interested they become in history, I think.
I agree with that. On a somewhat, although I must say that I was surrounded by those people when I was learning my parasitic diseases, and there's such a rich and unusual history associated with that I think I adopted that earlier than most people and shared that with my medical students, and it worked out pretty well. On a somewhat, thank you for that caveat. On a wonderful related note, several years ago, I did a project where I went back through 30 years of HIV research, selected 1 or 2 papers per year, mostly going by the number of citations, and then asked one of the authors to write a commentary on that paper, putting it in historical context. It's not exactly the kind of taking the paper apart that you discussed with Dr. Firestein, but some of them are quite interesting. You can find all these commentaries here, and then he gives a link. And if you want to read just 1 or 2, here are 2 good choices, and he gives two papers that he lists on the internet. Best regards, Jaeger. P.S. Vincent, if TWiV were a paper, would you be the first or the last author?
I don't know. I have no idea.
What would you be?
I guess the last author.
The last author.
You'd be the senior author, clearly.
I think you would be.
I don't know who'd be the first author. Depends on the episode, right?
Right. We had Harold Varmus as the first author last episode, I think.
He would be, yeah.
Absolutely.
Yeah, if TWiV were a review article, then you might still be the first author.
Right.
We'd do it alphabetically.
Good thing it's not.
and given
alphabetically, how would that work?
Given that we will now have 401 publications, I think we could rotate.
Interesting. And in Britain, they do it by alphabet. They
okay.
Alphabet.
And some disciplines do it by alphabet.
Yeah, that's true. Physics, for instance.
I'm obviously fine with an alphabetical approach.
I would be delighted.
Yes.
I include a little history in my virology course, but I do get the sense that they're not really all that interested unless it's really stunning, something really stunning. So
right.
I keep it to a minimum.
I mean, Jenner's discovery, how could you that out?
Stuff like that. I tell a little story about transformation by RNA viruses, a little bit here and there, just because I want to introduce people. I think people make
that's right.
Science.
Yeah, but then how many? Do we have to know these names?
Yeah, I know.
Right.
Yeah. I always preface.
Is this going to be on the test?
I say no. You don't have to know these names. I just wanted you to get a sense for, what happened. Yeah, they asked me that too. Do we have to know these names? Oh my gosh. Yeah. Sorry, the Firestein episode is so long ago now that I don't remember. But I think we did talk about using history to drive a course, but maybe it's not such a great idea.
Yeah,
well, a history course.
A history course of
but not,
science course.
I think a science course it needs to be organized more conceptually, just because of the sheer volume of information that you need to communicate to catch people up on everything that's been done for the past 500 years.
But it does depend on who presents it. I mean, this series that I mentioned, I think a long time ago, called Connections.
Right.
This guy Burke, he did such a wonderful job of using illuminaries throughout history to connect the ideas that led to unexpected discoveries. I really appreciated that show very much.
All right, the next one's from Steve. We'll do one more round, okay?
Yeah.
Okay.
Hi, Vincent and fellow gender-bewildered Twivsters.
I'm not sure that came out quite right.
Yeah, I have no doubt about my gender.
I'm not gender confused.
But we understand what he's saying.
Yes.
I sympathize with your problems trying to find the right or acceptable forms of address for people, both singular and collective, but recently read this being discussed on the BBC site in regard to the various forms of gender identity that are being recognized these days, and discovered that there is already quite a movement in U.S. university circles to get new non-binary pronouns accepted. So you are far from alone. Personally, I don't think that allowing people to specify their preferred pronoun really achieves much, because it still leaves people who don't know them guessing. However, for collective terms of address, as in trying to replace your guys, a reasonably reliable term ought to be findable, though the BBC article implies that none of the efforts to establish terms to date has yet caught on. I had thought that perhaps we ought to go down the taxonomical route and try homos or sapiens, homes, saps, but those already have confusing uses. Incidentally, guys hasn't, in my experience, been considered derogatory in England, but until recently, someone using it might have been considered a sude for putting on American airs. That's great. I think it would mostly be accepted now, though. From reading the BBC piece, it rather begins to look as though the non-binary replacement for you guys is likely to be you theys. Looks odd at first, but nowhere near as confusing as the individuals who now want to be addressed as they in the singular. Parens, actually thinking again, you've already coined your own terms that will work on your podcasts, at least. Twivsters, twipsters, etc. People suggest new ones to you in their letters every week. Personally, I think that people attach far too much importance to their sex lives and sexuality in general, and do wish they would actually find something really important to get angry about. It's not as if we're short of impending catastrophes. Anyhow, you might like to read and post this link to the BBC piece, and you could possibly bring in a spokesperson from one of the unis that are trying out the new pronouns. And he sends this very interesting article in the BBC News, Beyond He and She, the Rise of Non-Binary Pronouns. And these are for people who, don't identify as he or she anymore. And what do you call them? Transgender, cisgender, non-binary, all this. Very interesting. I sent them.
And all the pronouns, and I especially find that using the they and their for the singular to be very ambiguous when it's written. And then, in fact, in one of them, they gave an example, and they have to follow it up with a parentheses to explain that they mean the individual person. So it includes University of Wisconsin-Milwaukee, Ohio University in Athens. So a couple of places that,
At the University of Vermont, which has led this movement, students can choose from he, she, they, and ze, as well as name only, meaning they don't want to be referred to by any third-person pronoun, only their name. Wow. Keep up the good work, though I have to admit that I'm finding all this genomic evolution stuff very difficult to follow. You could for a start, usefully explain how long a base sequence has to be before it is unique and can only belong to or have come from one species. All right, we'll do that on TWiFo.
Yeah.
Stephen Hawkins has to ask a question like that.
Yeah, isn't that funny, Steve Hawkins? It's Hawkins, not Hawkings.
Yeah, right.
Completely steel gray overcast at the moment, but at least not windy. All right. Now, Kathy can take the next one. I just want to say this is also about Firestein, and Neil wrote this to Stuart and also mailed it to TWiV, and I thought it had some points that were of interest to our audience. That's why I suggested we read it. I'm sorry it's so long. You can paraphrase at points if you wish.
Okay. So Neil writes, Dear Stuart, this past weekend I listened to the latest podcast in which you participated. I enjoyed the discussion overall and appreciated your willingness to devote your time and effort to activities beyond the securing of your latest grant or publishing your latest original report, which, after all, are necessities to keep one's position. I'm an immunologist and clinical pathologist with major interests in such areas as immunity to pathogens, transplantation immunology, genetics, and the relevance of evolution for medicine. Since my undergraduate days, I've also maintained a strong interest in logic and aspects of the philosophy of science, especially the philosophy of biology. So I wanted to provide a bit of feedback. In general, I guess that our perspectives are reasonably compatible. I agree that the scientific method is, in quotes, is largely a fiction. That said, I do not accept that, as you say in your TED Talk, I'm not a fan of TED Talks, science is really just farting around in the dark. I do not question that experimental inquiry always proceeds in the presence of uncertainty and incomplete understanding, parentheses, metaphorical darkness. But what scientists do, at least successful ones, is not usually just random fooling around in the lab. Some ignorance does not correspond to total ignorance on any level. factual, methodological, theoretical, or conceptual. As you argued, in noting the overselling of the role of serendipity, scientific discoveries are usually made by scientists with extensive prior education, training, and experience in experimental or observational investigation. Therefore, I think you are overselling your point about ignorance, at least in the TED Talk. And I'm just going to make a little insertion here. Chance favors the prepared mind comes to mind.
Yep.
Okay. Back to his letter. By the way, my antipathy to TED Talks is multi-pronged. The atmosphere is worshipful with respect to the speakers, which is antithetical to the sort of somewhat critical attitude to speakers that is most appropriate in a scientific context. Second, and related to the preceding point, there are no questions. Third, there's a real temptation to distort the truth for cheap laughs or a bit of extra drama.
You are not alone in your opinion, Nick.
Here, here.
Mm-hmm.
Having given one, I can attest to it.
I have a lot of respect for a lot of the people who have given TED Talks, but I don't think that the format is all that great.
No, it doesn't allow for detailed explanation of anything.
Right. It encourages a certain leveling of nuance that I don't think is a positive thing, especially these days.
Right, and there's a certain amount of showmanship involved that also
That's right.
makes it artificial. Or,
the E is entertainment, so they have to emphasize that a little bit.
Right. Okay, in the course of the podcast, there were many comments, too many to keep track of, that made me want to respond, so I'll just cite a few. Regarding Ray Kurzweil, I think he's deluded. And, below is a link to a commentary I published on his expectation of living hundreds of years. So he's got that. With respect to Thomas Kuhn and his extraordinary influence, I think his framework for how scientific revolutions proceed is massively flawed. One point is that he extrapolated to all of science from a very small number of examples, primarily from physics. Biology is quite different. Although I haven't written an article, I hope to eventually write one analyzing the discovery of MHC, major histocompatibility locus restriction, a major advance in understanding of how T cells recognize antigens, to explore how this revolution fails to conform to Kuhn's rigid conceptions. Arturo Casadoval and Ferric Fang have recently carried out a fairly thorough analysis of what they hope are representative scientific advances and find that these discontinuities in understanding do not generally conform to Kuhn's rather rigid scheme.
If you want to criticize Kuhn, you can get in line.
Okay.
There's a substantial history of criticisms to Kuhn. So.
Okay. This discussion of pitchers aiming for outside the strike zone went a little off target. Pun intended. Okay, I added the pun intended. Okay, of course, pitchers need to tempt batters to swing at pitches outside the strike zone, but it cannot really be denied that some situations require aiming for the strike zone. Certainly, fastball pitchers will sometimes try to throw at maximum velocity right to a batter's sweet spot to demonstrate dominance. With the bases loaded in a close game, I expect some pitchers will intend to throw strikes, at least for some of their pitches. Okay. The notion that lifespan extension has not created problems yet is clearly debatable. Look at Japan, or consider the long-term outlook of Social Security in the United States. The absence of complete social disruption so far hardly demonstrates that there would be no problem if average human lifespan was 100 years. I have no doubt that I would enjoy discussing an indefinite number of topics with you, but I do not make the mistake of presuming absolute symmetry. Nevertheless, if you do wish to respond, I would be pleased to receive your thoughts.
An interesting
regards, Neil.
Interesting way to say that, presuming absolute
symmetry. Yeah.
Right.
You may be more interesting than me.
Right.
All right.
So do we know if Stewart replied to him?
I don't know. No, I didn't ask him. I assume he might have, or maybe not. Stewart tends not to reply to me, so I'm not sure.
And Neil, by the way, is a professor of pathology at Case Western Reserve.
Right.
All right, Alan, you're next.
Okay, Kim writes, "Hello, Vincent. You've been mentioning crowdfunding on the podcast now for quite a long time. I've been listening for a long time, and your podcasts have influenced me a lot in my day-to-day life. The quality of the content you and your co-hosts make is astounding. I really would love to contribute to the podcasts in order to help you produce even more amazing content. Thus, I urge you to create a Patreon page so that all the people who love your podcasts can contribute and give back. You will be amazed by how many will want to contribute, and you're missing out on a lot of funds every month if you keep procrastinating on this topic. There are various entertainers, such as YouTubers, who make huge amounts every month via Patreon, and similar sites with content much inferior to yours. So please, Vincent, get with the program.
Nice.
I just wanted to put this up. Go ahead. Sorry.
Was this the inspiration for finally getting around to starting a Patreon now that we have one?
Yeah. Go ahead. Okay. You were going to read the last line.
Yes. Best regards, individual who is feeling more and more like a thief for every Twix podcast he consumes.
So, yes, I wanted to include this to thank Kim for pushing me. I finally did it. And, of course, we do have a Patreon site, patreon.com/microbetv. At the moment, we have 67 patrons.
Great.
And, our goal is lofty. My goal, I put up a goal of $5,000 a month, which would allow me to hire someone.
Aim high.
Allow us to travel and so forth. None of us are going to pocket this money.
No.
We're going to use it. So I think we have more than 67 listeners, but I'm patient. I understand. And if you would like to support us, go over to patreon.com/microbetv.
And if you are one of those 67, thank you very much.
Thank you so much.
No question about it.
Indeed. At some point, I'll put a list of them all up. But we really appreciate that. And you can give anything from $1 on up. And, $1 a month is not much for 5-6 different podcasts or however many I would like to make. And we appreciate it. So thank you, Kim, for stimulating that. And Dickson, take the last one.
Okay. Dave writes, "Hi, I really appreciate your show, but I didn't think I would ever have anything to write into you about, as I am not in a scientific field. However, I came across this game, which made me think of you guys and lady guys." Thank you for that genderizing. Called Incel VR on Steam. Now, I haven't looked at this, so I can't honestly say that I know what it's all about. Has anybody on our team seen this?
Yeah, it's an interesting game. You need VR glasses, virtual reality glasses to play it, but go ahead and...
right. They also have links to iOS and Android versions on their website, but the Steam page has a video. It involves...
The iOS version would work
regular.
with
that's right.
the Google Cardboard, right?
Yeah.
Yeah, or maybe you don't need anything.
Right. It involves racing through a cell trying to beat an invading virus to get to the nucleus, and has some interesting visuals. It brands itself as a VR game, but a VR headset is not required, and I do not have one. It is also free to play. I'm curious what you think. Thanks for all your work creating an informative and entertaining show.
I think it's pretty neat. I've run through it, and the graphics are neat. They've taken some liberties as usual, but they're pretty stunning, and it looks like fun. The real test, though, I will ask my younger son to play it.
So you played it, Vincent?
Yes, I have played it. It's good. Of course, I'm not a gamer. I am not a gamer.
Did you do it on your iPhone or something?
iPhone, yeah.
And did you use the Google Cardboard?
No, I didn't. It doesn't. You don't have to have a VR device. No.
Okay, because we got one of those with the New York Times a couple of months ago, and
really?
Yeah, they sent it out for free. They had this, of course, being the New York Times, they used it to bum people out. They had this virtual immersion story on the Syrian refugee crisis. So they put you in the shoes of a refugee, and it's like, oh man. But
yeah, it was a Sunday, and you got your Google Cardboard, and
Neat. No, you don't need it. I did it without. But if you have it, try it, Alan.
Okay.
Let us know what you think. I mean, the visuals are nice, as he says, really cool. But as far as the gaming environment, I'm not a gamer, so I'm going to let my young guy do it, and although he'll probably say, I don't want to do this. We'll see. I'll let you know. All right, let's do some picks of the week. Dickson, do you have a pick? Or I should say, what is your pick, Dickson?
Well, I had one.
You had? You lost it?
I did. No. I put it up. It was the Juno probe, and it hasn't actually given much in the way of results yet because it's still finding out which orbital platform to use before it swoops down to within 2,500 miles of the surface of Jupiter, taking pictures all the way. So I'm going to pick that when it happens.
Oh, okay. So you're not picking that this week?
No, I'm not picking that one. So I found another one.
So you have a non-pick. Okay.
I have a non-pick
This is something new, a non-pick of the week.
A Rosamund Purcell is the artist, and she makes constructions using art. And she claims, the claim is that art and nature are the same. And when you look at her constructions, you might agree with that. They're bizarre, and they're unusual, and they're inventive and creative, and very, some of them are kind of spooky, but others are just evocative, or they elicit feelings of pattern recognition. But they're strange. They're very strange, and they're very artistic, and they're very beautiful, some of them. I don't like all of them, but then again, you don't have to.
I'm looking through these pictures, and Donald Trump's daughter shows up.
Yeah. Well,
Why?
I didn't do that.
Why?
Don't ask me. Ask the artist.
Oh, my God. Yeah, these are interesting.
They're interesting.
I'm not sure that art is
interesting.
nature, but it can be.
Yeah, well.
It's fine.
Okay.
She's entitled to her opinion, right?
I like visual things. So that fit into my...
Thank you, Dickson.
You're welcome.
Cool.
Alan, what do you have?
I have a band called the Double Clicks. And I thought I might have picked a song by them some time ago, but I searched the TWiV site, and I guess I didn't. They are a very nerdy duo, and they're proud of it, and it's awesome. It's two sisters, and they do songs about really nerdy stuff. So, Star Wars and Star Trek and comic books and this sort of thing. They had a quite an internet hit a few years ago. That was forgetting what it was called. I'm looking on their YouTube channel and not immediately seeing it. But if I come across it, I'll highlight that. Anyway, if you go to their YouTube channel or their website, you can get some samples of their music. The one they've got leading on their YouTube site right now is called Cats at Parties. And it's about being an introvert and going to a party and just, going to find the cat to hang out in a side bedroom with the cat while the party's going on.
Yeah, so as soon
Kathy.
as Alan put this up, I've heard them live. They sang in a small venue here. My brother knows about them and had heard them before. And the room that they played in is smaller than the room we recorded in last week. So
Really?
So it was a very intimate audience.
So
yeah.
change into the single clicks?
Well, no, I looked at on their website. It looks like they I missed a chance to see them a little while ago. They came to Northampton, Massachusetts, and they played at a comic book shop there. And I gather that's a fairly standard thing for them to do, is to play the comic book shops.
Does John Schaefer know about these people?
I don't know. But anyway, they're definitely worth checking out. They're a lot of fun.
So they have a Patreon page.
They have a Patreon.
They're doing very well. They have 556 patrons.
That's
and
nice.
I got an idea from them. They have a big Patreon button on their website.
Then.
I should do that.
Yes.
Imitation is the highest form of flattery.
No, it's good to get ideas.
The hit that they had 3 years ago, it's got over 1 million views on YouTube.
Wow.
The video is called "Nothing to Prove."
Nice. I like the banana. I like the banana costume. That's
yes,
cute.
the banana costume and the giant cat organ are apparently regular features these days.
All right. It's good to get ideas from other people, right?
Yeah.
What do they say? It's good to imitate others, but not yourself. Someone said that. I forgot who it was. Kathy, what do you have?
So, first of all, Kathy writes, I forgot to mention last week that the bioartography site has other formats for the art for sale, not just the note cards that I gave you guys. And I forgot to mention that the proceeds go to fund student and postdoc travel to meet.
Oh, gee whiz.
Very good.
Nice.
I remember that with Alan and Rich when we were up at the cloisters. So I decided that's how I would do it. So I picked two Zika-related things. One is a WHO Zika app. And I had trouble when I went to the App Store and tried to download it. So then I opened the email where I had this linked, and then clicked through that way and got it. But anyway, it's a nice WHO Zika virus app. It's mostly meant for medical providers. I have to admit that I learned at least one fact from it, and so it's just kind of a nice handy thing, and I think they're going to keep it updated. So check that out. The second thing is that a former student of mine was at the ASMQ conference in Washington, and as swag, they got a number of things. One of them was a Zika virus giant microbe, and she texted me the picture of it when I was in New York last week, and then she texted me pictures of the Washington Zoo, and I texted her pictures of the Statue of Liberty. But the main thing was that she sent me a picture of her Zika virus giant microbe. And then later, when I got back in the mail, she had gotten another one and sent one to me. So there's a Zika virus giant microbe now. They came out as of April 20th, 2016, so they're very current. And the site that I linked to, which is the regular giant microbe site, has a thing on it where it's not just a fun thing. If you buy a giant microbe, part of the proceeds go to an entity called Direct Relief. And then if you follow through on Direct Relief, there's something called biotraps. And I looked at the biotraps thing, and it looks really interesting. And they've tried it in Australia. And so I wanted some people to look at it and see what you think of it. I don't know how vetted it is, but the overall gist of it is that it's a paperboard bucket that has a treatment that allows it to persist and be water resistant for a while. And you put a female mosquito attractant in the bottom, and then this inside part of this bucket is lined with larvicide, and you put water in it, and it attracts the females, and they die because of something that's on the side, and the larvae that she lays die. And then just about the time that the larvicide is going kaput, supposedly, is when the paperboard thing disintegrates, and so you're not left with standing water.
What do you think?
A neat idea.
What do you think, Dickson?
It's worth a try. Sounds just crazy enough to work.
So, and supposedly if you buy one, they'll donate one, and I don't know. So anyway, mostly it's because if you look at this giant microbe of Zika virus, it doesn't look anything like a Zika virus.
It doesn't.
Right.
Usually they make them look
I'm
like
looking at something from heartworm that Vince is looking at. It doesn't look like heartworm either.
Yeah, the Zika does. It's got two Z's on it and two I's, of course.
And it's yellow and green, and it's
right.
not like the
they're meant to be cute, I guess.
It's not right, I tell you.
Dixon, I found heartworm for you.
It doesn't look like a heartworm to me.
Doesn't?
No, heartworm is a nematode, for gosh sakes.
It's very cute, though.
It's not cute. I don't think it's
the
cute at
Zika
all.
virus has knobs and stuff. I'm just here.
By the way, why doesn't Kathy? Why doesn't Lego make a set of viral construction Lego?
Oh, that would be cool.
Oh, that would be cool. Ah.
We should suggest one.
We might.
Can you still do that sort of
You
suggesting
can
thing?
suggest it, then you have to crowdsource it. You have to get 10,000 signatures. It's a pain in the you-know-what.
Forget it. I'm not getting 10,000.
You have to do their marketing research, which is, I think, wrong.
Right.
Well, that's pretty cool, though.
Yeah.
All right, thank you. My pick is a PBS documentary that just aired 2 nights ago. Did you watch it, Dickson?
I did watch it, actually.
I did too.
Oh, lovely. Spillover.
Yeah.
Yes.
Zika, Ebola, and Beyond.
That's right.
It is...
Is that based on the book?
No.
No, it's not based on the book. But this is produced by Tangled Bank Studios, which is a production studio bankrolled by HHMI. Right? So if you go to Tangled Banks, the video itself is very nice, lovely visuals, and well done. And Tangled Bank Studios is an HHMI-supported movie production studio. It's pretty cool, and it is somewhere in Maryland, I think. Chevy Chase, Maryland. And they basically will partner with filmmakers and make productions such as this one. They've done Your Inner Fish with Neil Shubin, Vaccines Calling the Shots, Mass Extinction, Life at the Brink. So they do nice work. And so there it is. This is on PBS, and it's good. I don't know how you see it again, though, if you want to, Dixon. What do you do? Do you know?
You record it.
Yeah, which I did because I knew I was going to fall asleep, and I fell asleep partway through it.
How did you record it, Kathy? On the TiVo or something? How did you do that?
I'm really a dinosaur. I still have a functioning VCR.
Nice.
Really? Wow.
Yeah.
Save it. It's worth a lot of money someday.
Yeah, it's worth
No, but on the website, you can order the DVD.
I see. Okay, so you can't just see it anytime, right? They don't do
Well,
that.
a lot of PBS stuff does get played over again. So if
Yeah.
you
It does.
have a functioning TV guide, you could probably figure that out. But our paper stopped carrying one for free, and I'm not going to pay for one.
Anyway, it's worth looking at. Check it out. And good production values. Well done. Nice pictures of. I love the bats. They have great images of bats, which are so cool.
They truly
Check
are.
that.
Yeah.
We have a couple of listener picks. The first one is from our listener Ricardo, who writes, "Hello, TWiV fans. I finally catch up with the late episodes. Project Premonition was a wonderful idea for an interview. What we can do with great minds, money, and the wish to make good. On the last episode, Dickson's weekly science pick was about Germany nearly reaching 100% renewable energy. From the 7th of May till the 11th of May, during 107 hours, Portugal electrical grid was supported 100%
Look at that.
by renewable resources."
Let's have a contest.
And he gives a link to that 4 days straight.
Yeah, that's great.
Always a pleasure to listen to you. GU ... Sorry, Kathy. All. To listen to you all.
The thing is, I say guys. I grew up in the Midwest. Guys is everybody. I do not take offense at
Hey,
it
you have a brother.
at all.
If you have a brother, everybody's a guy.
I don't know.
Hey, guys.
It just doesn't bother
No,
me.
I think group is a good substitute for that. Okay. Group,
Over
listen
on
up.
TWiM, we had a lot of discussion, and your colleague there at Michigan wrote a long email about it. So, and it was a guy who did that.
I'm
Get a
still
lot of-
lobbying for you all, but really.
All right, another pick from a listener, Fatma, writes, "Dear TWiV, I want to bring to the listener's attention a novel bioinformatics tool recently published in Bioinformatics that has the ability to predict virus-host protein-protein interactions. This can be of help for many virologists working on human viruses and want to limit their research space when it comes to find which human proteins are able to interact with their virus proteins. I'm one of the researchers on that work. This does not make it novel or less useful." And I will be
Or more
happy,
fundable.
I will be happy to help any research group interested in using that tool. We are already helping different labs across the world, and I hope will be of more help to the scientific community. And he or she links to the paper. And Fatma is a PhD candidate at Virginia Tech,
Cool.
which
Place.
we were just at. Maybe you came to ASV. And finally, Stephen sends a link to a YouTube video, How Mosquitoes Use 6 Needles to Suck Your Blood. And he writes, "I now like them even less."
Right.
I love mosquitoes. They're so beautiful.
They're well engineered.
They're very annoying, though.
It's annoying, but I happen to be one of those people who's never bitten by mosquitoes. And someone next to me can be covered with welts because I'm objectionable. I like mosquitoes. They're amazing. I mean, look at them. They're so beautiful. They have evolved in such a cool way. How can you not? Yeah, I understand, but I love viruses too, and we all do. This is TWiV. You can find it at iTunes and microbe.tv/TWiV. Please send your questions and comments to twiv@microbe.tv. Kathy Spindler is at the University of Michigan in Ann Arbor, Michigan. Thank you, Kathy.
Thanks. This is a lot of fun.
Dickson Despommier is at verticalfarm.com. Thank you, Dickson.
You're welcome.
Father of the vertical farm.
You're welcome.
You hate when I say that.
Oh, I've learned not to hate.
And as long as you hate it, I will say it.
I love it when
Because
you
you're
say
my
it.
friend. Because you're my friend.
Keep saying it, Vincent. Keep
Alan
saying
Dove is at alandove.com. He's also on Twitter as Alan Dove. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. I would like to thank today's sponsors, CuriosityStream and Drobo, for their support. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.